Good afternoon to all, and good afternoon to our speaker, Dr. Andre from State Key Laboratory for Planetary Science, Lunar and Planetary Science from Macau. Thanks for accepting our invitation, Dr. Andre. We look forward to hearing from you deep sea exploration and learn more about astrobiology. So, welcome, and the stage is yours.
Okay, thank you very much. First of all, thank you for the introduction and for giving me the opportunity to participate in your webinar series. I've been following up on this. I've checked some of the previous guests. I think it's a really interesting event that you have here. I'll start by sharing my screen. Let me see if this will work well. Okay, so I think you can see my screen now. Okay, so in today's talk, I'm going to talk about terrestrial analogues in astrobiology. My name is Andre Antunes. I work in a couple of different fields. Just let me minimize this. Yeah, I think now it's better. Okay, so I'm an associate professor and leader of the astrobiology unit at Macau University of Science and Technology, and specifically at the State Key Laboratory of Lunar and Planetary Sciences. Feel free to look me up on Google Scholar or ResearchGate. I'm also on Twitter. And I know that we have a Q&A session after the talk. But if you want to do some additional follow-up, or you're feeling camera shy or something like that, I'm more than happy to answer any sort of question or queries that you might have. Okay. Regarding the type of research that I do, most of the work that I do is linked with the exploration of the limits of life, particularly biodiversity, and also a little bit of biodiversity of marine and extreme environments across the globe. Done a lot of work in locations in Africa and Europe. I'm keen on starting doing some research also in Asia now. I've only just recently moved to this part of the world. I work with geomicrobiology, interactions between microbes and minerals, which is a really interesting cross-disciplinary field, and work with the development of new bioinformatic tools, databases, and new lab techniques. Strong suit of my research at this stage is work linked with astrobiology and also connected with planetary protection. I'll discuss a little bit of this later on. And I work with a combination of bioinformatics, microbial cultivation, and characterization, together with some molecular methods, omics-based approaches. Okay, so for you I'm not really sure how many of you are familiar with Macau, if you've heard about Macau before or not. So Macau is an amazing city. It's a special administrative region of China. It's a former Portuguese colony, and it was returned to China in 1999. It's really a wonderful place to live in, a unique combination of modern and old Asian influences and European influences, and it's a really nice place. Regarding my institution, so as I explained, I'm in Macau University of Science and Technology. It's a relatively recent institution. We've just turned 20, and we've been doing really well. Our campus has an amazing location right next to the big casino area of Macau, which Macau is famous for. We've been doing really well in expanding and investing in research and getting better in rankings, international rankings. It was just announced earlier this week that our institution was placed in number 38 globally in young universities, according to the Times Higher Education World University Rankings. We also have special connection with China's National Space Administration, which is really handy. It's really useful for the research that we do over here. And so, starting with the talk, I am a microbiologist by training, so I work with microbes, and I find them amazing. I think that they're really cool and interesting organisms. And so we owe a lot, of course, to some pioneers, and probably the most important of all is this guy here, Antony van Leeuwenhoek. He was the person that is credited. He's sort of the big father of microbiology. Was the first person to observe microbes and do the first drawings of microbes, and he used this very rudimentary tool that he adapted. It was sort of a rudimentary magnifying lens that was a little bit more powerful. It was a precursor to microscopes that we use. And the reason why he had this specific tool was that his family worked in textile industry. And so they used gadgets similar to this to analyze the quality of the textiles. And because he was very curious and interested about what surrounded him, nature, he liked putting all sorts of samples, all sorts of things into this gadget. And so that's how he realized that that there were small organisms living everywhere. And of course, the technology that we have nowadays has evolved drastically. We're able to get a lot more detail, a lot more information on the morphology of microbes, and partly based on information that we get from electron microscopy, for example. We're now able to differentiate between prokaryotes and eukaryotes. And eukaryotic cells are cells like our own and the ones that all animals and all plants and fungi have. Cells that have nucleus, have a well-defined nucleus. This is in contrast to prokaryotes, which are simpler cells. They have a simpler organization and have no defined nucleus. DNA is just sort of loose inside the cells. We do have a big difference. We do have a big split within prokaryotes between bacteria and archaea. And this is a more recent development, although it's been a while. It's been around for a couple of decades now. We owe it to this other pioneer. This is Carl Woese. He died in 2012, and he is a very, very important researcher in the field of molecular microbiology. He was the person that was responsible for our current view of the tree of life. And what he proposed was to make use of techniques that were brand new back then, and looking at sequence of DNA and comparing DNA sequences as a way of mapping out and tracing the phylogenetic relationship between different organisms, so that we were able to see whether two different species were more closely related or further apart, if they diverged a long time ago or only recently from an evolutionary perspective. And the big breakthrough there was his suggestion to use a specific gene that was associated with the synthesis of a specific type of RNA, 16S RNA. And so this 16S RNA is a vital component of ribosomes. And ribosomes are essential piece of cellular machinery. They're responsible for synthesizing all proteins that we have inside cells. And so it's based on the analysis of this gene, which in our case is slightly different, but it's equivalent. And so it's the analysis of this specific gene and the comparison of the DNA sequence that allowed us to get this sort of visual representation of biodiversity on our planet and the creation of the so-called tree of life. And so the tree of life, as we currently understand it, is composed of three main branches. One composed of eukarya, where we fit together with all animals, all plants and fungi, for example. And then there are two additional branches, one for bacteria and one for archaea. This is not a static view. There's ongoing developments, and there's actually a heated debate nowadays whether we should merge the eukarya branch with the archaeal one, because there's actually a lot of things that we have in common with archaea, as opposed to what was originally thought, because they're quite simple and do resemble bacteria in a variety of other aspects. And so the the development of these molecular-based techniques completely changed our perception of life on our planet. We now know that we live in a microbial world. Microbes dominate our planet, clearly. They are the dominant life form. And they're not only the dominant life form on our planet, they're even dominant. They're even the dominant type of cells in our own bodies. Estimates point to us having 10 times more bacteria in our bodies than our own cells. So this is just an amazing thing. And even more mind-boggling are these numbers. Current estimates point to a number of bacterial cells in our oceans that is much, much higher than the number of stars in the universe. So that's just something unbelievable. So they're incredibly abundant and incredibly diverse. And there's a lot of things that we still don't know. In addition to this high abundance and high diversity, microbes are incredibly versatile, incredibly flexible, and incredibly resilient. They've been able to populate and thrive in all types of extreme environments in our planet, from low pH to high temperature to alkaline conditions, low temperature environments. Microbes do really, really well in different types of extremes. And of course, my very favorite is composed of high salinity environments. And so microbes that live under these type of conditions, they don't just survive extreme conditions, they love extreme conditions. That's the ideal type of environment for them to do well, and that's where they feel happy and do well. And so organisms that live under this type of extreme conditions are called extremophiles. Of course, we need to be a little bit careful with definitions, and it's always a little bit tricky. And so defining extremophiles as organisms that love extreme conditions is a little bit human biased. It's a human-centric perspective, because what we perceive as extreme, for them, it's normal. Okay? So we need to be a little bit careful there with how clear these definitions are. And so there are different types of extremophiles dependent on the different type of extreme that we're considering. And so when we're specifically discussing high salinity environments, we call these types of organisms, we refer to them as being halophiles. And so halophiles do really, really well. They thrive under high salinity conditions. And if you live in if you live or if you've had contact with coastal environments in warmer, sunnier climates, you've probably seen this sort of landscape before. So this is common solar saltern. So this is a common way of producing salts. And so it's very normal for you to see this sort of pinkish, reddish, orange coloration associated with several of these ponds. This color comes from the extremely high density of microbes that do really well under these types of conditions. And so regardless of the fact that these conditions are actually quite harsh, salinity is very, very high, we do find representatives of all three domains of life in this type of environment. So we have Artemia, so we have brine shrimps, we have Dunaliella, and we have both bacteria and archaea, some of them with really unusual shapes, like this middle green one that you have here. That's very, very common inhabitant of high salinity environments. And people struggled studying it and isolating it in the lab for many, many decades. And of course, the special environment that I work with and that I want to discuss in a little bit more detail today is the high salinity environment, but it's a little bit more complex than that. I work with deep sea brines. And so deep sea brines were originally discovered purely by accident in the Red Sea area in the late 1950s. There was an expedition, a Swedish research vessel that was collecting samples from the bottom of the Red Sea. And one of the samples, when it was brought up to deck, when people were checking the conditions of the sample, they were surprised because the sample was very warm. In addition to that, it was very, very salty. And this was something that was completely unexpected. People didn't really know, people didn't really understand what was going on. And so we now understand a little bit better where this comes from. And so what we have is that the Red Sea is opening up. So it's a sea, but it will eventually become a fully formed ocean. And so you have the splitting between the Arabian and the African tectonic plates. And this tectonic rifting that is currently occurring creates several different depressions, deeps at the bottom of the sea, more or less along the central axis of the Red Sea. In addition to that, before the Red Sea existed, in the same geographic region, there was a prehistoric sea that dried up completely, and it eventually formed a thick layer of salt. So underneath the Red Sea area, we have this thick layer of salt. And so when you have tectonic rift process occurring, it creates cracks in the Earth's crust, and it allows regular seawater to trickle in, to have contact with the salt, partly dissolve it, and then it resurfaces as brine, water with very, very high salinity. Because brines, because of their high salt content, they're much denser than regular seawater. They tend to stay in deeper areas. So they quickly fill these isolated deeps that I mentioned before. And this is what created the deep sea brines of the Red Sea. After the original discovery of the Red Sea brines, of the original Red Sea brine, there was a big survey that was done across the Red Sea. There were some expectations that this might be linked with oil, which wasn't actually the case. And so we now know that there are at least 25 different deep sea brines in the Red Sea area. In addition to this tectonic rift process and enrichment in salt, the fact that you have this active tectonic process taking place also ensures that the seawater that resurfaces as a brine is also comes at higher temperature and is enriched in a variety of different salts, ions, and metals. And so as two different examples here of deep sea brines of the Red Sea, we have Kebrit Deep. And Kebrit Deep is one of the northernmost ones, and it's one of the smallest ones. It has a total area of around 2.5 square kilometers. And because it's one of the most recently formed, that's why it's still small, the level of hydrothermal activity, the hydrothermal input has been somewhat limited so far. So the temperature is colder than in other brines. It's still good temperature. It's still increased when you compare with regular seawater. So the temperature in Kebrit Deep is around 24 degrees Celsius, close to 24. On the other side of the scale, we have Atlantis 2. Atlantis 2 is one of the southernmost ones, and it's one of the oldest ones. So it has a much bigger scale. It has a massive scale, around 40 square kilometers, and temperature is much higher. It's the hottest deep sea brine that is known on our planet. Temperature is currently around 68 degrees Celsius, but there's been a constant increase in temperature since its original discovery. So temperature is still going up. After the original discovery of the deep sea brines in the Red Sea, there were a couple of other locations, similar sort of settings. And so there were a couple of other places on our planet that were found also to have deep sea brines. And we have some examples here in part of the Mediterranean Sea, and also a few isolated cases in the Gulf of Mexico. They all have in common this sort of tectonic activity associated with an area where prehistoric sea existed and dried up. So it has to do with exposure of the salt layer. Despite their common origin, despite the fact that they're formed in a similar way, each deep sea brine has a distinctive physical-chemical signature. There's chemical variability. Brines are different from each other. And that has to do with input from local geochemistry, from local geological setting, and variations in the Earth's crust and also in the salt deposits. So we do see some variability there. And of course, the deep sea brines that we find in the Mediterranean or the Gulf of Mexico are more divergent than when you're comparing across the Red Sea. An additional thing that is really interesting about the deep sea brines is the fact that because brines are so dense, as I mentioned before, they don't really mix with seawater. They have very limited mixing occurring with seawater. And so you have this very sharp transition zone, this drastic change in conditions from deep seawater to brine. So at the brine seawater interface, you have significant increase in temperature depending on what brine you're analyzing. You have transition from oxygen availability to completely anoxic, massive increase in salinity, of course, and then you have change to completely different type of chemistry. As an additional component there, because the brine is so dense, all sorts of particles that would normally sink throughout the water column all the way to the bottom, when they reach the brine seawater interface, the majority of them are not heavy enough to break through the interface. And so they just accumulate there. So the brine seawater interface is particularly interesting because it's a transition in chemical conditions. There's a heavy particle load, and it's a very diverse type of ecosystem. So it makes for a really interesting place to analyze from a microbiological perspective. And so depending on where you're sampling within the interface, you have complete different conditions and reality. As you can see here with the picture of these two filters and the concentrated, we concentrated the sample using in situ filtration, and you see that even the biomass that we accumulated there and the metals that accumulate there are different in color and in chemistry. That really reflects how drastically the conditions change from one depth to another. I have here some video footage that I wanted to show you. So we were lucky enough in one of the latest expeditions that we had to the deep sea brines of the Red Sea, we were able to bring an ROV with us, and we got the first video footage from the deep sea brines of the Red Sea. You will see increase in particle load and the camera entering the brine now. Okay, so you see this thick particle load, sort of whitish beige coloration, very, very heavy load. We do see some microfauna that seems to benefit from this high particle load over there. And then we saw this really unusual additional feature. This is video footage from Kebrick Deep. And as we were moving the ROV across the surface of the brine seawater interface, we were able to visualize these very unusual red patches. We're not really sure what this is. It seems to be some sort of biofilm-like structure that was disrupted once we moved a little bit closer. And so we're not really sure what it is. It could be that it's likely that it's linked with differences in microbial populations that live there, and it might be linked with a specific group of microbes that do really well in high salinity environments, as we saw with the salterns. Okay, these are really amazing video footage that we got from this location. Okay. And so how do we study the deep sea brines? This is a recurrent question that I have. It's the thing that people most commonly ask me. So how do you get the samples? Deep sea brines are located quite deep. The shallowest in the Red Sea is around 1,500 meters. The majority of them are usually around 2,000, 2,200, up to 2,400 meters. And so we need these big research vessels. And we use a combination of different tools that are common in oceanography. So we use things like rosette samplers that you see here on the left side. And they're very handy tools. They're not particularly complicated. They're quite simple. It's basically just a hollow tube with two lids, one on top and one at the bottom. And you lower it. It's connected to the ship, and you lower it to the specific depth that you're interested in. It comes equipped with all sorts of sensors, so we're controlling from the ship, and we're able to pinpoint exactly what depth is it at, what's the salinity, what's the temperature, what's the oxygen level. And once we reach the depth that we want, we can trigger the closure of the lids. And so we're successfully trapping water from that depth within the tube. And we can trigger different bottles at different depths or multiple ones at the same depth. We just bring it back to the surface, and that's how we get our samples. To get sediment samples, we normally use things like this, multi-core. This is a specially adapted multi-core unit. It has duck feet, and adapted on the spot. Had to do with samples from one of the sites where the sediment at the bottom was really, really soft, and we have the multi-core kept on sinking deeper into the mud. So we had to adapt this thing, and we got really good samples out of this. So that's good. Yeah, so this is what I explained with the closure of the Niskin bottles and how it works. Okay, so regarding microbiology of the Red Sea brines, it's a really interesting story. So I have the original papers, the first papers looking at microbiology of these sites actually say the brines are sterile. Nothing can grow there. Conditions are too extreme. No type of organism would be able to survive high heavy metal concentration, high pressure, high salinity, in some cases high temperature. It was just considered too extreme. And of course, there were some limitations. This was in the 1960s. They relied on just the classical approach, just with regular plating and seeing if anything grew. So might have not been the most appropriate approach, but it was the standard thing that people did back then. It is nowadays we use a very different approach. It's common to use a combination of different things. We collect data from the environment. We use cultivation and isolation of microbes, but we also combine it with molecular-based approaches, namely metagenomics and omics approaches in general. The first phylogenetic studies that looked into the deep sea brines of the Red Sea came with very interesting results, very surprising results. And here I'm showing you a tree. Don't worry too much about what's there or what's written on the text. The important thing for you to notice here is these are results from two different brines, one marked in red and another one marked in blue. And what we were able to get out of these first studies was that we completely dismantled the idea that nothing could live down there, or that if anything would be down there, that the brines wouldn't be very diverse. It was a big surprise to confirm that despite the combination of multiple extremes, these brines were teeming with life. They had a very diverse population. There were very different groups of microbes living down there. Some were specific to one location. Other groups seemed to do really well across multiple brines. And most important of all, they were completely new to science. They were unrelated to, they didn't really have close relatives. They weren't really related to, that closely related to other known microbes. So they were completely new. And this was true both for bacteria and for archaea. Further studies done more recently and done in the Mediterranean, now moving more to the use of metagenomics, have further increased the number of new groups that were identified from deep sea brines and confirmed that there were a couple of specific microbial groups that seem to be adapted to deep sea brine conditions and do really well, so that they were present both in the Red Sea and in the Mediterranean across all deep sea brines that we looked into, which was something that was really interesting. In addition to that, the use of these molecular-based techniques allow us to confirm that there was stratification of the microbial communities. So, as I explained before, because the conditions in the brine seawater interface change so much, it's not completely unexpected to see drastic changes in the microbial populations that match this. More recently, we had some of the very first insights into the viral diversity, the viruses that live down there, and we were able to show that there was a high level of novelty of new viruses living at the brine seawater interface, and seemed to be particularly interesting setting and particularly interesting conditions in Atlantis 2, which is the one that is more extreme, where you have the higher temperature conditions. And we also showed that the viruses also had this typical stratification where it was different if you were sampling higher in the brine seawater interface or lower. Okay, so regarding microbial isolates, what did we get from there? What new microbes were we able to get from there? So as I explained before, these were the original results: empty plate, nothing growing. I already explained that there were some limitations with this type of approach. Not that we shouldn't keep on using it, but we should complement it with additional methods. And this is one of the highlights that I wanted to show you here. One of the techniques that has worked really well for us in isolating new microbes is the use of the optical tweezers technique. The optical tweezers makes use of a laser beam, and you have here a combination of, you have here a culture of of microbes. So you see several different cells, and what you have is you're coupling a microscope with a laser beam that you can see here with a cross. And what you do is you can activate the laser beam to trap the cell in the beam, which is a really cool thing to do. Not just because we're using lasers and lasers are cool, but the fact that you can actually mobilize one individual cell. You can isolate one single cell out of a mixture of different ones. And so this is a really handy technique for separating one cell from a mixture of cells and getting pure cultures, which is a vital thing in microbiology. For you to be able to study microbes fully and understand what their capabilities are, understand what they do, we need to get pure cultures. And so this is what we do. We separate the individual cell within a glass capillary. We cut it so that we're separating the one cell from the mixed culture, and then we use it to inoculate culture tubes and see if it grows. Okay, so it's a very elegant technique. It's a very handy way of isolating microbes that might not grow using standard techniques and regular Petri dishes and plates. Okay, before I give you a little bit more information on some of the species that we were able, some of the new microbes that we are able to isolate from this setting, from the deep sea brines of the Red Sea. Just wanted to give you this overview of taxonomy and different taxonomic groups. So, because I think that we're all familiar with species and what species mean, and you're probably all familiar also with kingdoms. So we're part of the kingdom Animalia, which stands for all animals. I will be referring to groups that represent new orders and new phyla. Okay, so when I mention a new order, that's equivalent to finding the very first carnivore. Or if I say that it's a new phylum, it's equivalent to finding the very first chordate. So it is a very big discovery. It's a very big addition to the tree of life. So the very first microbe that was isolated from deep sea brines of the Red Sea was this one, Flexistipes sinus arabici. Was isolated from Atlantis 2 deep from the brine seawater interface, and it represented a new phylum. So completely new. Later one that I got, that I isolated as part of my PhD, Saldinisfera shabanensis. And Saldinisfera shabanensis represented a new order. It belongs to the gamma proteobacteria, but it is a new order within the gamma proteobacteria, and it has a variety of different one of the things that I noticed straight away is how versatile it was, how flexible it was. It was able to grow well with oxygen, without oxygen, microaerobic conditions, low salinity, intermediate salinity, very high salinity. So it had very wide growth ranges for a variety of environmental factors, which fits really well with the lifestyle, with the fact that it was isolated from the brine seawater interface, where there are drastic changes in environmental conditions. So this should be an advantage for this type of for this organism. We also analyzed the genome, and we were able to showcase a couple of genes and a couple of enzymes that were associated with this variability in local physical chemical conditions, and also some features that would make it interesting for potential biotechnological applications, namely for bioremediation, so treating contaminated soils and waters, and detoxifying heavy metals, and this fits well with the conditions that it lives in. It was also able to produce bioplastic, which is something which is a topic that is becoming increasingly relevant and increasingly vital. Additional organism isolated from another brine seawater interface was Marinobacter salsuginus. In this case, it was just a new species, but still a very interesting organism. Had very interesting capability to degrade oil-derived compounds. And it was also able to oxidize iron. So it seemed to be linked with some sort of local geochemical cycle that occurs at the brine seawater interface that involves iron. And an additional organism that has a really interesting story is this one. These are photos that I took from a culture that I had, and there was a vacation period coming up or something, or long weekend. I don't really remember the details that well anymore. I left cultures in the incubator for longer than expected. I completely forgot that they were there. And once I remembered and I checked them, I saw that the colonies looked strange. They have these weird inclusions, these weird round shapes on top of it. So all of these, so the background that you see is the surface of the colony, and the round forms that you see are the structures that I saw. We did some studies. We didn't know what this was. We had never seen this before. And we were able to eventually identify these as being biominerals. These were calcium carbonate crystals, which are really, really interesting. There's a bunch of biotechnological applications associated with them. You can ask me about it in the Q&A session if you're interested to know more. It also might have interesting implications on the ecology of these locations, because these crystals would be dense enough, they would be heavy enough to break through the brine seawater interface density barrier. And carbonates are known to absorb organic molecules, so they could play an important role in particle traffic from seawater into the brine. Another microbe that I wanted to highlight is Halorhabdus tiamatea, the first member of the archaea that was isolated from deep sea brines. For a long period of time, it was the only one. I think there was another two that were recently described from the Mediterranean. It has very widespread distribution, so similar microbes were detected in different brines of the Red Sea and the Mediterranean, and had a couple of additional physiological peculiarities that made it really interesting. But my very favorite is this one, Haloplasma. And Haloplasma has this very unusual morphology. And so if you work with microbiology, if you study microbiology before, you know that from a morphological perspective, microbes tend not to be very different from one another. You normally have one of these two basic shapes. They either look like spheres or they look like rods, like baguettes, long rods. And so whenever you find an organism that has a morphology that is not neither one nor the other, that's a big deal. That is something that is remarkable. And so cells of Haloplasma look like this. They have a central round body, and then they have these tentacles that coil and uncoil, which is something that is unique and something that has never been seen in the microbial world. So it has this unique morphology, very unusual contractility. And so we now know, because we analyzed the genome, that it has genes for cytoskeleton elements. So this might be interesting for biophysics, for example. And there's a lot of interesting questions that we still have about this organism. We don't know why it's doing this, whether it's just a way of moving around. It's a lot of things that we don't really, there's a lot of unknowns at this stage. We don't really understand it properly. We don't know what's going on or why. We also did, of course, some scanning electron microscopy studies where we saw that there were a couple of additional elements to this unusual morphology, namely these sort of filaments, some sort of filaments that connected the coils and some terminal blebs. So the morphology was a little bit more complex than we could see with optical microscopy. Fittingly, haloplasma had this placement that was between two big groups of bacteria, the firmicutes and the mollicutes. And it does share some common features with elements from both sides. So it might be some sort of missing link between these two groups. And we, at this stage, we don't really, we cannot be fully sure, but it does seem to represent a new phylum of bacteria, which is quite a big find in the current microbial taxonomy. Regarding a couple of further studies that I wanted to highlight, as part of the ROV operation, we were able to identify a new genus of mussels in one of the deep sea brines that we described, also completely new to science. And we also did some work associated with bioprospection, found some interesting activities against cancer lines, for example, and did some screening for enzymes that were interesting for biotechnology. And so for the final part of this talk, because, well, I think we're slowly running out of time here, why should we study extremophiles? Why should we care about this type of environment? And so, of course, on one side, you have this drive about applications, and a lot of people and a lot of funding agencies are very heavily centered on this. What can we get? What can we gain from this? And it is, people see extremophiles more and more as the solution to a lot of the problems, a lot of the questions that we have in the world nowadays, namely how to fuel the world, how to feed the world, and how to heal the world. In addition to this, of course, there are other questions that are maybe a little bit more sort of philosophical and helping to answer some of the questions that humanity has asked from its very beginning, namely, where did we come from? What's the origin of life? And so adding new branches to the tree of life gives us a better understanding of what the first cells on our planet looked like. And of course, this one particularly relevant reason to study extremophiles and study this type of environment is linked with astrobiology. And so it's nice to see that we've moved away from this sort of human-centric perspective. Sometimes I wish that we'd moved a little bit further than we have, but we moved away from this sort of human-centric perspective, thinking that we're special and the Earth is special and we're unique and there's no other place like this. We're slowly moving away from this sphere of ignorance, and so we are. There's more and more of a research focus on the search for life on locations outside our planet, and of course. The search for life has fueled a lot of ideas and a lot of people throughout different ages. And a lot of the discussion is centered on Mars specifically. Ever since the first observations of Mars from Schiaparelli and Percival Lowell that observed some sort of structures, they visualized some sort of structures that they thought were some sort of artificial canals this fueled people's imagination, and they thought that there might be some sort of civilization on Mars and little green men populating the red planet. And this has fueled a lot of the popular culture that we see from several decades ago up until today. The search for life got a big blow with the first missions that landed there, because there were such big expectations. And so when people realized that Mars looked quite barren, there was big loss of interest by the general public. This reversed back a little bit with the discovery of this specific meteorite, the analysis of this meteorite, and the announcements that were made that this could be evidence, this sort of microfossil could be evidence for some sort of microbial life on Mars, which is something that is highly debated and highly questionable. We know better nowadays that there are other reasons why we could have seen these microfossils. And so as I was explaining, we still have heavily centered discussion on Mars. Mars is not as barren as it seems. We do have growing evidence for the presence of brines on Mars in the subsurface, and maybe in some parts of Mars in certain seasons, in certain periods, we might actually have liquid water, salty water existing there. And this would be a condition for a necessary condition for the existence of life. The shift has moved a little bit further now to icy moons of the outer solar system, specifically Europa and Enceladus, because there's evidence for global scale oceans there underneath these thick ice crusts. So it's not really clear at this stage what type of chemistry, what type of physical chemical conditions do we have there, but we do know that these oceans are salty. And we do know that it's very likely that there's some sort of localized hydrothermal activity associated with these oceans. And this is valid both for Europa and Enceladus. We do see, and of course you've all seen footage you've all seen images of this, the majestic plumes of Enceladus. They do provide us a neat way of getting more information on the chemistry of these oceans. But of course, when we're discussing these exo-oceans, these extraterrestrial oceans, we're still several decades away from actually getting samples from these locations. So in the meantime, we need to center ourselves on looking at our own planet and looking at extreme environments. Extreme environments are ideal terrestrial analog sites, places that have conditions similar to conditions in other planets. And so if you look at the range of environmental conditions on our planet and the range of conditions where life exists, and you do similar mapping efforts looking at other locations on our solar system, it's not that unlikely that life could exist and thrive in different locations and outside our own planet. There have been a variety of different terrestrial analog sites that have been proposed and have been mapped. And they have a wide range of different conditions depending on what you are specifically interested in analyzing. Some are good Mars analogs, and some are really interesting for the similarities that they have from a geological setting, for example. And regarding the study of the icy moons of the outer solar system, discussion has been so far more restricted than the one about Mars. There's been a couple of terrestrial analog sites that have been proposed. Here you have an example, Lake Tires in Spain, due to the conditions that you have there. These are sulfide lakes, and it matches some of the expectations regarding chemistry of the oceans that you might have in Europa and Enceladus. And we also have some sulfur-rich Arctic springs in Canada in particular that do present some interesting sampling. So deep sea brines combine these characteristics. They do offer an ideal type of setting for studying life on the edge, life at the limit of existence, testing a variety of different equipment, and they really provide a very interesting analog to these conditions. I think, how are we with time, Bala? I still have time for this final section?
Yes, we have 5 minutes. No problem.
Okay, that's fine. That's enough for me to wrap things up. Just wanted to make sure if I could discuss this.
Certainly.
An additional reason why extremophiles are so relevant and are so interesting is directly linked with planetary protection. And I don't know if you've heard about planetary protection before. Most people that I know, when I mention planetary protection, they think of something like this. Some sort of superhero task team to save the planet or the universe or something like that. Actually looks a little bit more like this. Planetary protection normally revolves around this type of setting. And so, given what we now know about the resilience of microbes, we need to be very, very careful with missions that are sent to locations of our solar system that might have life. Okay? So the basic mandate of planetary protection is, on one side, to preserve the conditions for future biological exploration. We want to avoid contaminating sites. Because the thing is, if we're sending missions that cost a huge amount of money to find evidence of life on Mars or in the icy moons, we want to avoid a situation where we're bringing our own microbes with us and we get false positives, because then it's just a wasted mission. In addition to that, because our microbes, our extremophiles, do so well in all types of settings, they could eventually outcompete native life forms that might exist there, and they might cause collapse of entire ecosystems before we even know that they're there. That would be tragic. That would be a dreadful thing. That would be a terrible thing. The other part of planetary protection, of course, to protect our own planet. If we're bringing samples back from Mars, and we're going to do that soon, there are several missions planning this, both from initiatives taking place in China, NASA, now the UAE is also doing it. So there's more and more interest in bringing back samples from Mars. So it is important for us to make sure that we're able to protect our own planet from bringing something back that might affect life on our planet in some way, which is something that is of course, also very important. And so, of course, we have forward contamination and backward contamination. There's regular and ongoing discussions on rules and limits and how things should be handled to assure that we don't have these sort of contamination issues. And there's this rich cross-agency discussions to define rules and decide on what should be done and how things should be handled. As I mentioned before, extremophiles are incredibly resilient and they do really well. They survive exposure to outer space conditions. They survive all sorts of settings. And so there's more and more of an effort being done of submitting different types of extremophiles to exposure experiments, submitting it to radiation, to desiccation, exposing to Mars-like atmosphere. And so there's a bunch of ongoing experiments, including some with Salinisfera chabanensis that came from the bottom of the Red Sea. Experiments done with balloons. We're going to send this microbe to a scientific base in the Antarctic, and it's listed as one of the microbes that will be taking a trip going to the International Space Station as part of a couple of exposure experiments scheduled for 2023, if I remember correctly. Okay, so deep sea brines provide an ideal testing route for future research that is relevant to astrobiology, testing the limits of life. They're a source of new microbes, new microbial species, new strains that are ideal for testing and for exposure. They provide really interesting sites for looking at long-term viability and preservation, both of microbes and biomolecules, and developing and testing technology regarding sampling and simulation experiments that that are really relevant for future missions. So we started with us living in a microbial world and microbes being everywhere. I think it's becoming more and more likely that microbes, that some sort of microbial life could really be in a wide range of different locations across our solar system and elsewhere. And this would help us to answer probably the biggest question that humanity has ever asked: are we alone in the universe or not? Okay, so that's it. I want to acknowledge a variety of different institutions and funding agencies. I also wanted to advertise we're currently recruiting. We're looking for new faculty, new postdocs, PhD students, master students. If you're interested, just send me an email, ask me questions about it. We are a relatively new center and we're quickly expanding. And that's it. Thank you very much.
Thank you, Dr. Andre. There are a lot of questions that started halfway through the talk. So I'll read it one by one. These questions are coming up in the YouTube live chat. The first question is from Anurup Mohanty. What do you think of inclusion of viruses in the tree of life?
Okay, that's a very loaded question there. It's very complicated. So it's not an easy thing, and we need to be a little bit careful. We have this human tendency of organizing things and putting things in folders and structuring and creating walls where they don't necessarily exist. Viruses are not considered living organisms. I didn't go into the definition of life. That could be a separate talk altogether, just discussing what is life and what is not life. So viruses are currently not considered life, so they theoretically should not be placed in the tree of life. It would be complicated to fit them anywhere anyway, because they're incredibly simple. That's point one. They're not considered alive, but there are a couple of other things that makes it a little bit more complicated to discuss this. So some researchers think that they are a fourth domain of life, and they base their ideas on this, on the discovery that is somewhat recent of giant viruses. So giant viruses are a lot more complex, have much bigger genomes, and are a lot more lifelike than the vast majority of viruses that we know. So it's an ongoing debate. It's an ongoing discussion. But thank you for the question.
Yeah, a lot of questions. The next one is from Patnaik. What can we expect as analogues in the case of Europa in the perspective of exobiological?
What do you mean?
What can we expect as analogues in the case of Europa if you want to find life?
What can we expect as analogues? Well, deep sea brines, as I mentioned, are an ideal analog for conditions that you have in the exo-oceans of the icy moons. Regarding what type of life we can find, what type of life we can expect, if any, in Europa and Enceladus, it's very tricky at this stage. We still have very limited information. At this stage, we don't really have exact information on physical chemical conditions, but the fact that we have deep sea locations with hydrothermal activity and it's a brine ocean does make for some very interesting parallels with what we see in deep sea brines.
Okay, there's one question which is about life cycles. Again, it is from Anurup Mohanty. There are many extremophiles that are very tough, sometimes impossible to culture in laboratory settings. How do we study them and their life cycles?
Okay. It is one of the things that I really like, that I really enjoy, which is isolating microbes, growing them in the lab. It is, and it's the normal thing that the statistic that we normally mention is that out of the whole microbial diversity that we have in our planet, we've only been able to isolate and study less than 1% of the microbes that exist in our planet. So that's quite an incredible statistic that we have there. Regarding isolation of extremophiles and microbes in general, it is true that some of them are trickier to isolate, which makes it more complicated for us to study them. A lot of people rely on molecular-based techniques. So we analyze, we extract DNA from the environment, we sequence the genome, and we try to understand a little bit better what sort of capabilities are associated with these microbes. Sometimes you do find useful information to actually grow them in the lab. What I do believe, though, is that not enough effort is placed on developing new techniques, trying different things to get these microbes in pure culture and work with them in the lab. Because the shift has been to molecular biology, and everybody wants to do molecular biology. And the shift has been away from cultivation, and I think that we definitely need to invest more in it.
Okay, thank you. And the next question is from Manan Desai. What kind of impact does salt tectonics have on heliophiles found in deep sea brines?
What sort of impact?
What kind of impact does salt tectonics have on heliophiles?
I'm not really sure I understand the question. I'm not really sure I understand. So you have tectonic movements. You have the same sort of, well, you have slightly different settings, but you have them in the Red Sea, the Mediterranean, and the Gulf of Mexico. So in all of them, you have different types of tectonic movements associated with a thick salt layer. The impact that you have there is, and has been, that it allows seawater to dissolve the salt crust and create these new high salinity environments, which are ideal for halophiles. There's some interesting discussions, and I'm not really sure if it might be linked to what is being asked. There's some interesting discussions whether some of these microbial populations might actually be trapped in the salt layer. And so the tectonic movement would actually allow them to be released when they get in contact with seawater. So they're redissolved and released and become active and populate these deep sea brines.
Okay, I hope Anurup must be happy with the answer, I think. There's another question. It's very, very interesting. Would like to know more about the enzymes that help the microbes in the brine seawater interface to survive from the source.
Okay. There's a lot of studies that have been done on enzymes from halophiles. And halophiles are a very interesting source of enzymes that can be used for a variety of different purposes. We do have a couple of publications on enzymes that were specifically from the deep sea brines of the Red Sea that I recommend reading. Okay, but there's a very, very wide range of publications on different types of enzymes and different types of applications. They are very, very interesting for industry.
Okay, there's one question from Mohanty. Are bacteriophages found in such extreme environments?
Yes. So I sort of skimmed through it. It's always a little bit complicated when you have these sort of seminars, like you have a limited amount of time and you cannot really spend enough. Yeah, there's always things that people go like, oh, I wish that he would give more of a focus on this and you've given us more information. We do have, and I mentioned the viral diversity in the deep sea brines. The study that we did with metagenomics, we identified specific types of phages that live there, and they've been halophages, as they are known, have been detected in a variety of a very wide diversity of high salinity environments. So they are also present in deep sea brines.
Thank you. Next one is even much more interesting question from Matt. Should we target search on Martian lunar lava tubes where brine can be crystallized and safe from surface radiation? Can we expect preserved microbes there?
Well, the discussion on where to go first is an ongoing one. There are several different locations that are potentially interesting. Definitely the place to go is underground. Check the subsoil. And there's a couple of specific locations on Mars where there's evidence for, and I showed a picture of it, a video footage of it, where there seems to be certain periods, certain seasons where there's some sort of outpouring of brine. These would be interesting locations to look into also. But I think that future missions definitely need to dig into the subsurface.
Mm-hmm. Okay. Another question on the existence of life. The existence of life would require specific living conditions on different celestial bodies. What would be the Goldilocks condition for existence on Mars for the life we are looking for?
Okay. I'm not really sure if I got the end of the question, but so regarding habitability and where we can expect to find life and the whole Goldilocks area, the whole Goldilocks discussion, it's something that we need to be careful again. It's very difficult for us because we're heavily biased historically, and not just historically. The fact that we only know one place in the whole universe where life exists completely biases our discussion and our analysis. So the whole concept of Goldilocks, of a Goldilocks area, was very centered on conditions on our planet. And so it's now becoming increasingly likely that we might have life on places like Europa and Enceladus. And these would not fit within the Goldilocks zone. In addition to that, the whole discussion on where life is feasible or not feasible needs to take place at a more physical, chemical level, and not always necessarily based on what we know, based on the life as we know it. Because it could be that in the near future we find life on Mars, and there are two options. Option one, it's very similar to the life that we have on our planet. It might be even linked and fit within the same tree of life because there was movement of biomolecules, and there might have been traffic of microbes between both planets. And so that's option one. Life exists and it's very similar to our own, but it could be that life exists and it's completely different. And if life is very different, we need to be which is a possible scenario, we need to be a little bit careful with the assumptions that we make.
Okay, thank you. Do you have 5 minutes for a couple of more questions?
Yeah, sure.
Okay. All right, there's a question from Akila. How extreme the extremophiles, you can say, or extreme conditions will impact on human life?
How what, sorry?
How the extremophiles will impact on human life?
Will impact on human life? A variety of different things. So extremophile, well, in addition to helping to solve some of humanity's biggest questions, where did we come from and whether we're alone or not in the universe, and they provide really important insights there. They are used for a very wide range of things, from producing biofuel, bioplastics, new enzymes, new drugs, cleaning up the environment. One of the really interesting applications of biominerals, I mentioned biominerals earlier today. One of the interesting applications is its potential use for CO2 sequestration, a way of reducing the levels of CO2 in our atmosphere and helping to control climate change. So it has massive impact.
Okay. So another question from Das. Why is the temperature high and even getting higher in the deep sea, in the deep parts, like the Atlantis 2D?
Atlantis 2, okay. That has to do with hydrothermal activity. The tectonic rift process that is happening in the Red Sea. The Red Sea is opening a little bit like a fan, like this. And so in the southern part of the Red Sea, this process started earlier. It started longer. It's been active for a longer period of time. So hydrothermal input has been more pronounced. The deeps that, as you go further north, the deeps are relatively new, and hydrothermal activity and tectonic activity is less pronounced. And that's the reason why they're colder, and the ones in the south, you see this more pronounced activity and increase in temperature.
Okay. As another question from Karmakar, are brine pools found in Antarctica under the ice shelf? Are there any in situ studies in Antarctica on the brines?
There are a variety of lakes, of different lakes in the Antarctic. They don't share similarities with the deep sea brines that I mentioned today. I do know that there's, I think, hundreds of different lakes with different conditions. There's ongoing efforts to explore them and study them. They do offer some really important insights, particularly with how can you get a probe into these lakes through the thick crust of ice and avoid contaminating these pristine lakes that exist underneath it. So they do provide very interesting testing grounds, that's for sure.
Okay, that's another fantastic question. I mean, I like this question a lot. I'm sure you know. From Mehta, deep sea brines vary chemically from geographical location to location on Earth itself. How can we use them as analog to outside planet? Okay,
well, we can use them as analogs to conditions in the icy moons of the outer solar system. And the reason why I defend them and suggest them as good terrestrial analogs is at this stage we have several different pieces of the puzzle on physical chemical conditions in the moons of in the oceans of the icy moons of the outer solar system. And so according to some models, you have alkaline conditions, other models it's neutral, others it's acidic. And so in some models and based on some pieces of information, it seems like you have certain types of salts as more dominant than others. So it is important at this stage where we don't really know yet for sure what are the chemical conditions. It is good for us to have this diversity available. So the fact that we have diversity in the deep sea brines is interesting from a microbiological perspective because you will see different types of microbes in each one of these places, but it also provides proxies for different types of conditions. So we're able to use them as proxies for very different types of models and different types of possibilities regarding physical chemical conditions in these exo-oceans.
Okay, there's a question which is slightly related to this, research from Kamarkar. How is this research on brine pools impacting the study of spectroscopy for exoplanet atmosphere? Shall we or should we still search for oxygen-rich atmosphere and the signature of chlorophyll, or anything else?
Okay. Well, I don't have that much experience with these spectroscopy. I am a little bit concerned sometimes when I hear people saying, "Oh, chlorophyll, we need to look for chlorophyll," or "We need to look for this specific thing." Even when we look at our own planet, chlorophyll isn't the only type of pigment. There's some really interesting papers by Shilda Sarma from the US. He's one of the people that defends and he's a big fan of this purple Earth hypotheses. And so microbes that live under high salinity conditions use different types of pigments. They don't use chlorophyll. They're able to produce energy using sunlight, but they use different types of pigments. And so several researchers defend that this purple pigment that they produce might have been more important in the primordial Earth than chlorophyll is. So regarding the use of spectroscopy and looking specifically just for chlorophyll or just for this or just for that, I understand the advantages of focusing and looking for one thing, but I think you should always be careful not to exclude other options and other possibilities, particularly if you have evidence for that based on life as we know it, because that is known.
Okay. So in general, there is a question from Mohanty. What do you think did life originate in normal conditions and then move and adapted to extreme environments, or the opposite?
Okay, hard to say at this stage. A lot of people defend that Luca, the common ancestor that we have, was likely a hyperthermophile. So there are a lot of people defend that life possibly started associated with hydrothermal vents, and so the first life forms were extremophiles. And so what happened next was that organisms eventually evolved to less extreme conditions. That would fit with some of the ideas that we have about what the original, what primordial Earth looked like. Although there's it's an ongoing discussion. There's also the snowball Earth hypotheses. There's evidence that supports that there was a period of the evolution of our planet where conditions were very, very cold, and so it could be that the tree of life as we see it might actually be an offshoot of that period. So it's a little bit complicated. It is very important for us to add new branches to the tree of life to get further insights, but it's a very complicated topic.
Okay. I think we'll stop with the questions.
Okay.
Yeah. So thanks a lot from the organizers, from the students who are there. And there is a message from Dr. Vinita. She has sent this message in the YouTube chat. It was a very interesting talk to know about the deep sea brines. She thanks you for the talk as well. Thanks a lot on behalf of our students, our colleagues.
Yeah.
Thank
you very much once again for the invitation. Thanks for all the people that attended, and for all the students, and for the interesting questions. As I said, I'd be more than happy to follow up on additional things. If people want to send me an email or some sort of follow-up message, I'd be happy to continue discussions online. Okay?
Thank you.
Okay, thank you. Bye-bye.
Bye.